Throughout this video, we will be focusing on the blood-brain barrier. We will be examining its composition and function, as well as how this confers an evolutionary advantage. We will also take a look at some of its drawbacks, specifically with regards to the administration of medication. Next, we will turn to a discussion of some current research findings about the biochemistry of the blood-brain barrier, as well as the implications of this research. Finally, we'll ask you some questions and test your understanding of the material we've discussed. The blood-brain barrier is created by tight lacing of capillary endothelial cells with protein formations called tight junctions. These tight junctions are essential proteins that force all molecules to be transported from the capillaries through the endothelial cells, allowing for regulation of all substances that can enter the brain. Additional physical and functional support comes from pericytes, cells that wrap around the capillaries, as well as the end feet of astrocytes, which are a type of glial cell that connect with the cerebral capillaries. The tight junctions that we just described are an essential portion of the blood-brain barrier, and so I want to go into a little more detail about them. They are primarily composed of a branching network of sealing strands, and these strands, the more strands that they are, the greater efficiency of the junctions in preventing the passage of molecules. And these sealing strands are primarily composed of a row of transmembrane proteins embedded in the plasma membranes of adjacent cells. So the most abundant transmembrane proteins that are present are claudins and occludins. And these claudins and occludins create an association with different peripheral proteins on the intracellular portion of the plasma membrane. And this association serves as an anchor that joins together the cytoskeletons of the adjacent cells. In vertebrates, the blood-brain barrier serves as a specialized endothelial barrier that regulates the passage of certain chemicals from the blood into the brain. Similar to the plasma membrane, hydrophobic molecules such as caffeine, nicotine, and alcohol can easily pass through the barrier. On the contrary, hydrophilic and polar substances have a difficult time crossing the barrier and will sometimes need a transporter. An example of this is L-DOPA, which is used in Parkinson's treatment and is hydrophilic. This drug crosses the barrier using an amino acid transporter. Due to the molecular characteristics of amino acids, as explored in Chapter 3, not all amino acids can freely cross the barrier. In this case, amino acid transporters are necessary. It is approximated that 10 to 15% of all proteins in the brain's vascular system are transporters that facilitate the movement of substances across cells. An essential transport found on the blood-brain barrier is the GLUT1 capillary endothelial cell transporter, which helps with the movement of glucose. Overall, the blood-brain barrier is essential in conferring protection from pathogens and toxins by preventing them from reaching the brain. This serves to minimize the risk of damage. Additionally, the barrier denies entry of immune cells into the brain because while they are beneficial as a line of defense throughout the rest of the body, inflammation in the brain can lead to serious damage and loss of essential function. We can now understand why the blood-brain barrier serves as such an evolutionary advantage. It's because it acts as an additional line of defense between the brain and anything that would seek to harm the brain and could have possibly detrimental effects. This is a great evolutionary advantage, but it can also serve as a limitation in terms of pharmacology and the administration of medication. Because of the regulatory nature of the blood-brain barrier, scientists have to develop specific targeted treatments that can pass the blood-brain barrier and pass through the endothelial cells and through the tight junction so as to reach the brain in order to have the desired pharmacological effects to treat serious illnesses.
Now we're going to briefly talk about the environment of the blood-brain barrier, and more specifically what changes can occur in the environment and how these changes can affect the barrier. The first thing we're going to talk about is pH. Now normally the pH of blood ranges from 7.3 to 7.5, making it slightly basic but still in that neutral area. Changes in this pH can directly affect brain activity, and when blood pH suddenly drops, there's an increase in proton concentration, as we talked about in class, and this concentration is a result of the blood buffer system, where an increase in carbon dioxide will cause an increase in carbonic acid and protons. The end feet of the astrocytes that are part of the blood-brain barrier detect this increase in proton concentration in the blood and initiate nerve impulses using the surrounding neurons, and these impulses are going to send a message to the respiratory centers of the central nervous system, causing breathing to occur, and this will eventually stabilize the pH back to normal. And this particular phenomenon occurs when we hold our breath. That being said, so aquatic vertebrates that must hold their breath long have an evolved blood-brain barrier that can accommodate for sudden changes in pH. The next thing we're going to talk about is permeability and how trauma can affect it. And an increase in permeability can be caused by trauma or an infection where the surrounding tissues that are normally isolated from the immune system are exposed to lymphocytes, specifically B cells. And such injury or infection could grant pathogens access to regions that would be typically protected by the blood-brain barrier. And lastly, we're going to talk about receptors on the barrier itself, and receptors can cause an increase in permeability. Such increases in permeability can have potentially harmful effects on the body. One example of this is obesity, which involves a satiety hormone called leptin. Obesity in humans is the result of leptin resistance, and it is developed by a decrease in the level of the leptin receptor in the brain, or the saturation of the receptor that transports leptin across the blood-brain barrier and into the central nervous system. We'll shift our focus now to modern research. So we know that the blood-brain barrier plays a crucial role in the body, and with that, extensive research has been done for many years to further understand the barrier's abilities. A 2017 paper studied the therapeutic effects of a plant extract called curcumin and what role that it plays when a hemorrhage is induced in mice. And their goal of the study was merely to test the effects of curcumin on the blood-brain barrier once this hemorrhage was created in the mice. And most recently, Western blotting techniques have been used to detect proteins in different samples. And this method combines gel electrophoresis, specifically SDS-PAGE because we're working with proteins, as well as antibody binding. And the role these antibodies play is for visualization of the proteins for further analysis. The Western blot method was used to look at specific proteins that were involved during the hemorrhage and after the hemorrhage. In this experiment, the protein of interest was called MMP-9, which is a protein composed of both alpha helices and beta sheets, and it is involved in the degradation of the extracellular matrix. After the Western blot, the authors found that curcumin with the hemorrhage group decreased the expression of the MMP-9, which ultimately inhibits the degradation of the extracellular matrix of the blood-brain barrier. And a second method was also done, and it's often used in biochemistry, and it's called immunohistochemistry, or IHC. And this technique uses antibodies to visualize antigens, and in this study, the antigens were proteins, and it helps visualize proteins, similar to the antibodies in Western blot. The protein of interest in this part of the study was called IBA-1, and it detects microglial activation. And from the name, we can tell that microglia are a type of glial support cells in both the central and peripheral nervous system, and their job is to engulf debris in the body. Now for this, the authors wanted to visualize the expression of the IBA-1 to see what changes the curcumin induced on the microglia during hemorrhage. What they noticed was a decrease in the protein's expression. It was a decrease in the IBA-1, showing that there had been a decrease eventually in the microglial cells being activated. Based on these findings, they found that curcumin overall plays a large therapeutic role in hemorrhage, and that it can keep the integrity of the blood-brain barrier after a hemorrhage has been induced in mice for this study. As we've mentioned earlier, the blood-brain barrier is made up of these tight junction proteins, which contribute to the selectivity of the barrier. And remember the MMP-9 proteins that we previously mentioned and how that degrades the extracellular matrix? Well, it turns out that protein is also responsible for degradation of these tight junction proteins found in the barrier. The author's conclusion from this paper, from the Western blot of the MMP-9, showed that the MMP-9 expression decreased from the curcumin treatment after hemorrhage. And if that decreased, then the tight junction proteins wouldn't be inhibited, and they would still be translated and be part of the extracellular matrix, keeping the blood-brain barrier intact after the induced hemorrhage in the mice, further showing that the curcumin treatment after hemorrhage keeps the integrity of the blood-brain barrier.
The results of this research have a multitude of implications for the future and the future study of the blood-brain barrier. We believe that one major takeaway from this is that this research could forge the way to develop new treatments that can minimize the brain damage in individuals with serious head injuries. Additionally, this research could be taken one step further by analyzing the chemical compound of curcumin and trying to understand why it exhibited the effects that it showed within the paper, why it minimized the damage to the blood-brain barrier, and how these properties can be harnessed in order to be utilized medicinally in the future. Finally, here are just a couple of questions to test your understanding of the material and get you thinking a little bit more about the blood-brain barrier.